I'm Zhao Zhuo. I'm currently assistant professor in the Stevens Institute of Technology, and we're talking about how to perform like data-driven single-cell gene-gene interaction discovery, and we're proposing a new methodology called weighted diversified sampling. And because I'm non-bio background, so I'm just give a brief introduction. Like what we want to study is like how two genes interact and trigger some specific diseases. In this case, we studied the data with a focus on Alzheimer's diseases. Basically, we only study from the single-cell data how two genes interact and trigger the disease. And so we're saying like in the single-cell data, two genes are represented as features. So in the machine learning methodology, we say it's a feature representation in a single cell where every gene represents a feature and there's associated expression level regarding with it. So on the left, we can see there's a sparse matrix. Basically, there's a 1, 2, 3, 4, 5. There are 5 cells, and each cells may represent certain level of genes. And every cell has a different expression level. For example, for the first cell, the expression level is 7, and then, there's another gene expressed at 6. And in the machine learning perspective, we view it as a feature. Like we say, it is a sparse feature, like a recommendation system where everybody open Amazon, you buy Coca-Cola, you buy Pepsi. These are sparse features. And on the other side, like what we break it down is like there's scaling factor. Basically, your expression level of that genes, and also the indexes basically means what kind of gene are expressed in these cells. And then what we want to do is that we want to train a classifier. Given the training data, what we want to achieve is given a cell's single-cell gene expression, can we successfully classify whether this cell is infected by Alzheimer's disease or not, basically AD or non-AD. It's a binary classification problem. It's a simple machine learning classification task. And strong intuition of our methodology is like, suppose I have a perfect model. I have a perfect model that trains to successfully predict if every cell is infected by Alzheimer's disease or not. And there's a gene expression as features. Basically, I use this gene expression as a feature and do a 100% correct job and successfully predict whether the cell is affected by AD or non-AD. Maybe the model itself or how the computation happens in this model contains information about how two genes interact with each other to trigger this disease. So this is a data-driven way. It's like if I have a perfect model, I can do the job. Maybe the interaction within this model reveals something about it. And what I'll propose is like for one cell's gene expression, we mark it as a sparse matrix. Basically, every entry represents a gene, and we take the indexes as well as the expression level, and then we generate gene representation vector for every gene, and then we multiply with the scaling factor and pass into the transformer model. And here we want to highlight one strength of transformer-based model is the attention mechanism. So in short summary, what attention do is that they model they generate, for example, I have like 3 genes expressed in this cell. And what they do is that they will generate a 3x3 matrix where every entry of the matrix represents the interaction between 2 genes in the model. And what transformer do, basically transformer is the building block of ChatGPT and so many generative AI models. They build a lot of attention map in this model. And every attention map basically maps the interaction between 2 tokens. Here the token means genes. And we're not just talking about the single data. We're talking about multiple cells. And so we have to aggregate. And the aggregate is sparse because every cell is expressed with different subset of genes. So for example, here, like the first cell, they express this kind of genes, and we have to aggregate it to formulate a final aggregate interaction map. Basically, we'll pass through a massive data set with different cells, and different cells, every cell generates a attention map with the genes expressed in this cell, and we do sparse aggregation. And also we need to count the frequency, which find the frequencies also matters. For example, how many cells are frequently expressed in the Alzheimer's disease cells or non-AD cells. So by doing this, we get a final accumulate attention map. We call it a feature interaction map, like how two genes express from the data set itself instead of just a single cell example. And finally, this is the pipeline. Like we first train a model to classify whether it's a cell or not. Then given the one cell's expression, we generate the final attention map here to represent the gene interaction within this cell. And then we do sparsely aggregated to generate the final results. And then we can rank the feature interaction score from the N by N matrix. Basically, this N is representing the number of possible genes expressed in the data set, and we can take the higher value that as a ranking. For example, these two genes have a very high attention score across this data set, and we can show it as a strong indicator of these two genes are frequently interact to trigger the specific disease. And but we see another problem. So the foundation model are very expensive. They have a lot of parameters, consume a lot of memory, and one of the results is they cannot ingest a very large model. Basically, a large data set. You have a large data set, you cannot feed to the model by one time. So what we do for inference and training is we do batch-wise. For example, given a large data set, we do random sampling and do it step by step to reduce data. But one fun fact about gene work when I study is different from other applications. If you do random sampling on a large bioinformatics data set, you can see a lot of similar sequences. And that means this uniform sampling generates some bias during both training and inference. And we can measure it by Jaccard distance. You can see there's in bioinformatics, Jaccard is a very good distance. For example, we chop them into k-mer, and we measure the Jaccard similarity. And if we take a 100 example from a data set, we can see that their mutual Jaccard similarity is very high. It means that if you do random sampling over the data set, it's possible that you get a lot of similar sequences. And you are kind of introducing the bias into the model because the models keep ingesting similar sequences. And what we want to do is a simple trick is, because this is my previous PhD research, I focused on randomized algorithm. There's a simple hashing algorithm called min hash. What is min hash says is like, if two sequences are closer to each other in Jaccard distance with high probability, they will have the same hash code. So basically what we want to do is we take one pass of the data set. The complexity is linear to the data set. And then for every example in the data set, we generate a hash code. We can see that similar sequences are hashed with the same bucket. And then we take the counts. For example, the first bucket, we have 4 counts. And then the second bucket, we just have 1. So here we can see that a lot of similar sequences are falling in the same hash bucket. Then we take the reciprocal of the bucket and count as their weight. For example, the gene CCA is the only one in this bucket, means it's very unique. So we assign the weight 1. But for gene ACG, since there are so many similar stuff in the same bucket, so we assign the weight 0.25. In this way, we provide a cheap algorithm that go one pass of the data set, and we assign a diversity score for every example in the data set. It's a low cost. Of course, we can do expensive approaches such as clustering or something, but this clustering algorithm requires like n squared or more, like in terms of exponential regarding the number of data sets. We just take one pass and then we generate a sampling score. And then what we want to do is that once we have a model, given a large data set, we take a one pass hashing of the data set, and then we generate the score, and then we sample according to the weight to generate a subset. We find that this strategy provides an effective algorithm to handle massive data set. Like we select representative and diverse subset that still perform the same job. So the major intuitions to improve the data efficiency happen during the data-driven feature interaction discovery. And this is our pipeline. Basically, given original data set, we sample with the probability generated by min hash function, and then we select an example and take the attention map. as their feature interaction. Then we aggregate it because we're not doing uniform sampling. We can just not aggregate it with uniform weights. We have to also perform an important sampling formula. And for our evaluation, what we have tried is like we collect data set of both. These are ground truth knowledge graph for Alzheimer's disease gene-gene interaction. We can see how can we learn some gene interaction without prior knowledge, and how does it align with the existing knowledge about gene-gene interaction for Alzheimer's disease. And what we want to do is we take two data sets. First is how the gene is associated with specific disease. And the second is like how gene pairs interaction triggers the production of proteins. And we do a filter. Basically means that if one gene is strongly associated with Alzheimer's disease, and another gene is also strongly associated with Alzheimer's disease, and these two genes have a strong indicator to produce a protein, we say this may be our ground truth for evaluation. And then we take the enrichment score as an evaluation, and find the major observation is that with small sampling ratio, for example, we just sample 2% of the original data set, and we can perform a comparable performance with the original, which significantly reduce the data complexity in terms of the feature interaction discovery. So the summary of this strategy is like we want to improve the data efficiency because the model running both training and inference for the model is very expensive. And in the future, we'll also start studying how to build efficient model inference and efficient model training strategy. So these are something we're working on with Professor Zhang and Professor Hu. Currently, we want to build an infrastructure that enable. We see that from this project, we learn a lot. Like machine learning research generally don't care how the feature interacts and then trigger specifically something. They just chasing for like a better performance or higher accuracy or whatever. But right now we want to see how can we use advanced machine learning foundation models to trigger scientific discovery. So we can see it's not just gene-gene interaction. How about like protein interaction and cross-modality interaction? For example, gene and proteins interaction. And finally, we thank our collaborators from UT Health and also Rice University. We'll keep working on this project. Feel free to ask anything if you want. Thanks.
Do we have any questions?
Great talk. I think your research is really interesting. I really like the diversified sampling. That's a really great idea. I did have some concerns about the attention map. So I know you're talking about using a sparse matrix, but as we know that like the data is we don't know whether the zeros are really actually due to dropout, right, or they're actually zeros. And I can definitely see that affecting your attention map at least to some degree, right? I don't know if we have a way to actually quantify that, but I'm not sure that's like truly addressed by the attention map. Sorry.
Yeah. So that's a very good question. You're saying, for example, in the single cell data, there's one gene that don't express. For example, their expression level is 0. Does it mean like it's never expressed, or it does not help, or it ever help? So first, the thing is like if we just take all the if we take 0 value into the model, the value is actually not 0 because 0 multiplied by embedding is still 0, but 0 add the bias. So that's why the machine learning model contains the bias that to provide basic value. And another thing is like this is something is more open and I need to discuss with all the biological background researchers. Like if a single cell has 0 gene expression, does it mean that it does not contain this gene information or contain something? So if it does contain something, what is the contained level? Like if there's some level, we should maybe set some default value to that, or like we just mark it as 0.
Any other questions? All right, let's thank our speaker again.